Little bit about homeostasis and the mechanisms of maintaining it. At the top here, we have our definition of homeostasis. Right? There's sort of key words here. Maintenance of a relatively stable internal environment. Maintenance would indicate that this is an ongoing process and it's energy requiring. Relatively stable, as opposed to perfectly stable, means that it does vary. This environment does vary over time. And an internal environment, right? So the internal environment of the body. And as we describe it, the internal environment is the environment regulated in order for cells to survive. And so in our definition, the internal environment, if we come down to here to figure 1.9, is this interstitial space, which of course contains interstitial fluid. And we say that largely because all cells are surrounded by some degree of interstitial fluid with which they must exchange to survive. In other words, they have to exchange heat, they have to exchange nutrients and wastes and gases. In many cases, that exchange occurs with the blood capillaries shown over here on the right. Okay? So, what is the process or the main process of regulating homeostasis? Well, this internal environment, first of all, contains quite a, or encapsulates quite a variety of factors, right? Some of which I just listed. And so those physiological parameters have to first be sensed, right? And so the first thing that has to happen is we have to detect these particular parameters utilizing what we refer to as receptors. So there's a variety of receptor types for these specific parameters, whether that be blood nutrient levels or the waste levels or the gas levels, things like that. Right? And those receptors feed that information. They don't fix the problem. They feed that information into an integration center, often in the brain, but not always. And that integration center receives the receptor input, compares it to a set point, which is the point around which we are regulating that particular parameter, and would determine whether or not we are on or off the set point. In other words, whether we're okay or whether we're not. Right? And so here we have, in figure 1.8 over here, we have a factor, one of the physiological parameters varying over time, going up and down over time around this set point. And so the factors that make it go up or make it go down can be either internal or external to the body in many cases. For instance, generating body heat during exercise as opposed to entering a very warm or a very cold environment. Right? Both of those could alter body temperature. One from internal factors, one from external. Well, when we deviate too far from the set point, as we might up here or down here, right, we initiate a response to solve the problem, that deviation from the set point, to fix the deviation. Right? And so these arrows would be the actions taken to return our particular parameter to the set point. And so our receptor would sense those particular parameters. The integration center would determine whether we are or are not at our set point. And then if we are not, right, only if there's a deviation from the set point, we would actually activate effectors. And the effectors are the target tissues and organs that act to solve the particular problem. Right? So for body temperature, for instance, if body temperature is too warm, we might start to sweat or peripherally vasodilate, send more blood flow to our skin to release more heat to the environment. Right? Those are the effectors that become activated. Now these homeostatic loops, right, in figure 10 here, what we're showing is a homeostatic loop, are often called negative feedback loops because the actions of the effector, or effectors in some cases, will return our physiological parameter toward normal, right, toward our set point. And in that case, the integration center then would then read that receptor input as normal at the set point and shut off our effectors. In other words, negative feedback. The action of the effectors, the result of that is to solve the problem and turn off the system that was activated to solve the deviation from our set point.